resembles are Amelanchier, Aronia, Cateniaster, Malus, and Sorbus in the wide sense. These are all genera with, upwards, 20 to 200 species. The diploids are self-sterile and sexual. As with the Rosaceae as a whole, I guess, there's gametophytic self-incompatibility, and that's relevant down the line. In other words, seed set in these diploids depends on cross-pollination. Polyploids, however, can swing both ways. They can be reproduce asexually or sexually, and they have been shown to be self-fertile. In other words, the gametophytic self-incompatibility breaks down with the increase in ploidy level. Hawthorns, are notorious for having a really horrendous species level taxonomy that has been ascribed to hybridization, polyploidy, and apomixis, asexual reproduction. And that leads to questions about, if you have asexual reproduction, what are its consequences for the patterns of variation, especially in a group where taxonomy seems to be really problematic? Does asexuality get involved in hybridization? What's the impact of polyploidy? Do we have breeding system changes? Well, yes, apparently we do. And there are reticulate relationships. What's interesting is that for all its complicated species level taxonomy, there's a really robust infrageneric classification. And, there are subgenera sections and series into which most species can be pretty effectively pigeonholed. Well, the ones that I have experience with are. And a question arises of what kind of species concept is appropriate for genera like Crataegus. It's really important, where you are. I started out working with Jim Phipps at the University of Western Ontario in southwestern Ontario, and my PhD thesis research really was localized there in southern Ontario. The obvious comparison is with working with species that cross the entire continent. What was also important was there were a series of papers that came out around, either around the time that I started my work, and I've indicated those. I was not, flying blind. A lot of work had happened with different people, including my thesis advisor, and notably that paper by Rhoda Love and Mark Feigen in 1978 on hybridization between native and introduced hawthorns in Oregon. I should say it right now if I don't say it later on. Rhoda Love, the late Rhoda Love, was one of my mentors, a very important mentor who introduced me to plants of the Pacific Northwest. And then the other thing is that, as you'll see, the kind of technology that's available has changed over the period that I was working on these plants, and that's incredibly important. So this map is meant to indicate sort of the locus of my work on cockspur hawthorn, Crataegus cruscale. It's a group that was thought to include diploids at the time. it's incredibly, you'll see, I'll talk about how common it is, and here's an image of a hawthorn colonization on poorly managed agricultural land. Um, there are lots of hawthorns in southern Ontario, and I had the luxury of working at sites that really, were close to one another. I could drive around in a day and visit a number of sites. It was a pretty easy proposition that way. Um, just to note that the sites that I studied, for all intents and purposes, were all north of the glacial margin. And that will become important in a moment. But just let me explain what the rationale for studying these plants was. They have a very distinctive morphology. Their leaves are, very simple obovate shape, very shiny, unlike any other species in Ontario. They have long thorns, and at least in Ontario, these features make them really unmistakable. They're dirt common and locally abundant. I could study large topodemes, large local aggregations of the same species. And that was very important to me because I wanted to look at patterns of morphological variation within these, what people might refer to as populations. At the time that I did this work, people thought that there were both diploids and polyploids present. Work had been done on Apomixis in the genus by Phipps's postdoc Munyama, but there was nothing known about the reproductive behavior in the field. And I persisted in this folly when I moved to the ROM. I thought, well, um, let me continue looking at cockspur hawthorns and let me look south of the glacial margin. Can I find diploids in this area that was never glaciated? Well, I could not. Um, it was left for a graduate student who I'll introduce you to later on, to find the diploid cruskali in the south. The point was that this is an extremely species-rich flora, species-rich hawthorn flora in eastern North America, and trying to do the kind of work that I did in Ontario over this kind of geographic scale was a bugger. It really didn't pan out well. And instead, I turned to looking at the black-fruited Crataegus section Douglasii. It's disjunct in Ontario, and obviously widespread across the Pacific Northwest. It has a very distinctive morphology. You can't mistake the group in when they're in fruit. The fruit are purple black. And again, there was the surmise at the time that there were diploids as well as polyploids, that you could make some kind of a breeding system comparison in the group. And I say that reproductive behavior in the field was studied. Well, Rhoda Love was the person who did that. It was she who encouraged me to do field pollination experiments, and she shared her experience with how to carry out such studies. So I looked at Crataegus douglasii in the Great Lakes Basin. It's all through here, but I only looked at it in this part of Ontario. And then working with graduate students, we proceeded to begin collecting in Western North America. And at first, oh yes, there are diploids up in Haida Gwaii, in the Queen Charlotte Islands, according to the flora that was done. But, well, no buts. It's that was part of the reason for working on this group. Good documentation, supposedly, of diploids in the western part of the range. And just talking about this area here, you know, the distances between sites were considerably larger. And I'm sure for Western botanists, this is no surprise. I mean, what else, what's he telling us that's new? It turned out that the Haida material was not diploid. It is polyploid, much like Crataegus suxdorfii is all over. The diploids were found in Western Oregon and adjacent California and Washington. We also did work in this area here in the Rocky Mountains on section Saligny. But really, what I'm talking about is the series Douglassiani, and in particular, Crataegus suxdorfii and Crataegus douglasii to begin with. The important distinction between these is that Douglasii is a uniformly tetraploid with 10 stamens per flower. The Crataegus suxdorfii has the more primitive 20 stamens per flower that's found widely in the tribe Malei. And I was fortunate in having support from the Royal Ontario Museum where I was working. We had access to money for field work with which to build the collection, also to take part in mentorship and employment programs. I was also involved with the University of Toronto, and that enabled me to get very generous government grant support that allowed me to supervise undergraduate projects, but more importantly, graduate student and postdoc research. And as time went on, the tools got better and better. Got a good microscope. We learned to do flow cytometry. And eventually, as the right students came along, we started working with molecular methods. Just to remind you, the ploidy levels that we deal with are diploids, obviously, the ones that we have found. There are also triploids and tetraploids, and all of this variation in ploidy level has become much, much easier to document as a result of flow cytometry. Note also then that we also wind up discovering the contrast between allopolyploids, polyploids involving crosses between species, as well as polyploidization within species, resulting in autopolyploids. I'll skip this description. Flow cytometry is a well-known technique. It's becoming more and more accessible. People can use startup money to get a lab bench flow cytometer of their own if they're lucky. It's doable, and all of a sudden your sample sizes are huge. You're measuring many, many, many individual nuclei, admittedly from a single sample, but at least you can have some confidence in the results you obtain as compared with squashing chromosomes and so on. Flow cytometry of leaf tissue can give results like this, where you can contrast diploids with triploids with their somatic nucleus DNA content in this region here. There's a particular range of picograms DNA per nucleus that corresponds to the different ploidy levels. And then tetraploids are extremely common. And what we're looking at here is Crataegus suxdorfii and Crataegus douglasii again. Douglasii uniformly tetraploid, whereas suxdorfii, as it turned out, exhibiting both not only diploids, but also triploids and tetraploids. We also studied apomixis. This is the formation of seeds asexually by avoiding meiosis and fertilization. In the Rosaceae in Crataegus, this involves the formation of unreduced female gametes. Eggs are produced without meiosis, and these unreduced eggs develop directly into embryos by means of parthenogenesis. It's really important to note that endosperm fertilization may be required in situations like this for seed set to occur. Where it is required, that's referred to as pseudogamy. Why do this? Why do apomixis? One suggestion is that it was a Model T approach to evolution. Once you achieve a particularly good genotype, churn it out in huge numbers. On the other hand, other explanation was it's an escape from sterility, that if you have polyploids that may be sexually sterile, they can nevertheless fall back on apomixis to get their offspring out into the landscape. And does this eliminate genetic variation? Is this an evolutionary dead end? Well, where apomixis, as I've defined it, involves unreduced gametes, yes, you can clone yourself up with your unreduced female gametes and produce embryos and seeds without fertilization. Note, however, that in Crataegus, in other Malae, a pollination is required for endosperm to develop. But that's not all. These unreduced female gametes, these eggs, can be fertilized. You're using the plumbing of sexual reproduction, and guess what? You can all of a sudden wind up producing offspring with an increased ploidy level, and this is huge. But again, endosperm fertilization is also required. So you can imagine that what apomixis is doing is simply slowing down the tempo of genetic variation over time, but not eliminating it. I put the next two slides in case anybody needed to be reminded about what endosperm is, using the really blatant example of coconut meat and the tiny little embryo that eventually sprouts from a coconut. But of course, we're also familiar with exalbuminous seeds like peanuts, in which during the course of seed development, all the nutrients that were present in the endosperm have been translocated into the cotyledons, into the embryo. But in hawthorns, anyway, I can, and again, these other Malae, the seeds that are produced sexually or asexually, there's still a remnant of endosperm inside the seed coat. And it turns out that there's enough endosperm there that in a single seed preparation for flow cytometry, we can obtain signals for both the endosperm and the embryo. The critical thing, I mean, this is an illustration for a popular article that my colleague Nadia Talent helped prepare. Nadia Talent is the person who got Hawthorn flow cytometry to work. And she made a good thesis just out of doing flow cytometry and seeing what it would tell us about ploidy level variation in the genus at large and breeding system variation. Because in diploid sexual individuals, the endosperm nuclei contain 1.5 times the amount of DNA found in the embryo nuclei. Um, with apomixis, with polyploids that are reproducing by means of apomixis, the endosperm nuclei contain well over twice the amount of DNA found in the embryo. So it's a really handy tool for going and getting a quick idea of how these plants reproduce. And here's some results from that earlier sample that I showed you. This is from a paper by Eugenia Lowe. I feature her molecular work, which is really what was the focus of her PhD thesis at U of T, but she did masses of flow cytometry as well. And here you see the contrast between um, embryo nuclei and endosperm nuclei in diploids, embryo and endosperm in triploids, and embryos of tetraploids and the corresponding endosperm. So really, the flow cytometric seed scan is this amazingly powerful tool. This is Eugenia Lowe, and these are the sites that she sampled initially. And this is the result of an early microsatellite DNA study in which she summarized her results in terms of populations. These different labels, OR6, WA7, OR11, ID6, these are different populations of, in these open symbols, Crataegus suxdorfii, and in these solid filled symbols, Crataegus douglasii. And the shape of the symbol for the population indicates the ploidy level. So tetraploids have 4 sides, triploids have 3 sides, and the diploid populations have a single side. And note here that we could pick up presumptive allopolyploids according to this tree relationship in Montana and in Idaho. But in Oregon, we had what is presumptively an autotriploid showing up related to these diploids. Eugenia did some more work. The bottom line here is that using the microsatellite data to classify these populations, it became pretty clear that there were these 3 kinds of suxdorfii: diploids, autotriploids, and allotriploids, and allopolyploids. People have said, oh, well, hybridization isn't really all that common, but that was in the past. Um, we've shown, several people have shown that hybridization really does occur. And I put this in just to give you an example where these are Ontario taxa. And the important thing here is simply that these cells represent the frequency of species co-occurrence at a site. So the empty cells, Holmesiana and species 10, Crataegus pruinosa, occur at either no, 1, or 2, or 3 sites. They are uncommonly found together. On the other hand, species like Crataegus succulenta and Crataegus punctata are extremely commonly found together. So too, Crataegus punctata and Crataegus monogyna, the introduced European species. And we collaborated with the late Knolib Christensen to describe these hybrids as Crataegus X Nini chalatii to honor a former student who died. And the point here is that we're showing not just co-occurrence with the hatching of the cells, but this pseudo three-dimensionality here, this raised portion along the diagonal, also tells us something about the overlap of the flowering period of these species. And the co-occurrence data were assembled by Flemming Ulf Hansen, and Paul Smith is responsible for going and documenting the overlap of flowering periods. But for all that, the work that had been done was previously on hybridization, all had to do with diploids. And there was nothing about the process of allopolyploidization. Well, that changed first, starting with the work that Eugenia Lowe did that I showed you just now, where we have presumptive autopolyploids and allopolyploids, and then work by a postdoc, Mehdi Zarai. So now I want to just drop back for a moment and point out that these black-fruited hawthorns, they're present in the Great Lakes Basin. They're present in Western North America. And in Western North America, they overlap with red-fruited hawthorns in subgenus Americani. These are the subgenus Americani basically comprises all of the North American red-fruited hawthorns, notably Crataegus chrysocarpa and Crataegus macrocantha. Crataegus monogyna belongs to Crataegus subgenus Crataegus. The black-fruited ones that I was telling you about belong to Crataegus subgenus Sanguinii that occurs in Asia as well as in North America. We got lucky. We had the help of, we basically made the herbarium collection at the Royal Ontario Museum available to these barcode fanatics. And the results for Crataegus were pretty messy, but that led to Mehdi going and cloning one of these barcode loci, ITS2, and that gave us our allopolyploids. This is diploid Crataegus suxdorfii. Basically, all the suxdorfii-related taxa have this green color in them. Subgenus Americani, like Crataegus punctata, Crataegus chrysocarpa, Crataegus cruscali, Crataegus macrocantha, they're all shown in white. And the red represents the Rocky Mountain black-fruited hawthorns. And you can see that this is a good starting point for saying, yes, we have allopolyploids. And we can point to the way in which certain of the taxa that we studied share a maternal parent here. Similarly with the Saligny here. And then a lot of these things related to Crataegus douglasii have their shared parentage with subgenus Americani in this part of the tree. I got to keep moving. So black-fruited allopolyploids contain ITS2 ribotypes from both subgenus Sanguinii and subgenus Americani. On the other hand, The chloroplast tree that we were just looking at tells us something about the maternal inheritance, the directionality of hybridization. And as time went on, we have further additional and more comprehensive DNA data that corroborates the story of hybridization in these plants. So here's where we got to in Flora, North America. This was a treatment written by my former thesis advisor, Jim Phipps, in which he chose to say that the Pacific Northwest hawthorns in subgenus Sanguinii, section Douglasii, series Douglasii, all belong to Crataegus gay-Lussackia if they had 20 stamens per flower, or if they had 10 stamens per flower, they were either Crataegus douglasii or this segregative Douglasii with multiple thorns at a node, Crataegus castlegarensis. That was the simple approach. But the thing is, within this Crataegus gay-Lussackia, we have diploids, we have Crataegus gay-Lussackia sensu strictu of Marin and Sonoma counties that are autotriploids. They're also autopolyploids in Oregon at high elevations in the western slopes of the Cascades. And then there are all the allopolyploids in Crataegus suxdorfii. If you subscribe to the idea that species represent individual evolutionary trajectories, surely it makes more sense to think of the 20 stamen black-fruited things in North America, in California anyway, as belonging to, yes, Crataegus gay-Lussackia, the autotriploid, Crataegus suxdorfii, the allopolyploid, and we named a new species for the diploids in honor of the person who was my mentor when I was starting out, Rhoda Love. And here's some of the evidence to back up this argument. It's not just the reproductive behavior and the ploidy level. There are some morphological features that break out suxdorfii from Rhodomae lovii and gay-Lussackia, features of the thorns. The diploids tend to have thorns that are more slender at the base. The allotetraploid with 10 stamens per flower has the big thorns. The 20 stamen things have smaller thorns, shorter thorns. And as far as distinguishing between the diploids and their very closely related autopolyploids in Oregon and Crataegus gay-Lussackia in Marin and Sonoma counties, the gay-Lussackia has these tiny little petioles. And here's some work that was done by a graduate student, Jen Coughlin. I haven't shown very much of her work in this talk because her work was really focused on the ecology, the reproductive ecology, and the habitat ecology of these plants that she studied. But she documented their ploidy level. She documented them with microsatellite data. And this is just a summary of the way in which diploid suxdorfii, or Rhodomae lovii, and its autotriploid, are relatively similar genetically. They contrast with Crataegus suxdorfii, the allopolyploids, as well as with the allotetraploid Crataegus douglasii, which we surmise arose originally through allopolyploidization between 20 stamen black fruited things and 10 stamen red fruited things belonging to subgenus Americani. This is to give you an idea of the distribution of these plants layered over a climate map of Western North America. And here's Crataegus gay-Lussackia, sensu stricto, down here in Marin and Sonoma counties. But Suxdorfii and Rhodome lovii are present in Northern California. As it's We don't have a lot of data from here. We're pretty good for Oregon and Washington up into BC, and obviously into the Rocky Mountain states. But up around here, we really don't know very much. Here's the climate data. This is just taking bioclim data for the different sites that are represented on this map and looking at the constellation of climatic features. Here's Crataegus gay-Lussackia. It occupies a really different climate space from everything else. The diploids in Crataegus rhodome lovii are these open circles. And again, these circles are just found in this area here. It's the Crataegus douglasii and the allopolyploid Crataegus suxdorfii that are much, much more generalists. They have much wider distributions geographically and in terms of the climate that they can tolerate. So here's the alternative hypothesis. Allopolyploid Crataegus suxdorfii, autotriploid Crataegus gay-Lussackia, and diploid Crataegus rhodome lovii. In general, all three species are allopatric, but the data from Northern California is really awful. We don't really have a clear idea of how much these three species overlap. I wonder whether their ranges have been affected by human activity, both positively in some places and very negatively in others. But there's a lot of herbarium material, the Jepson herbarium, the herbaria at Berkeley, and the herbarium at Humboldt State, I know have lots and lots of specimens that could guide a student to go and do a lot more extensive sampling in the field. You could also sample from the herbarium material in order to go and get an idea of what's actually going on in Northern California. And is my hypothesis, worth holding up or not? Okay, and that's basically why I'm here. Is I wanted to go and tell Western botanists about this. I would have hoped that there would be some students. Well, if there aren't any students listening, people who supervise students, people who talk to students, tell them about this genus, tell them about the fact that there's all sorts of taxonomy to kick holes in if they choose by going and doing some work. Close to home. I mean, I had to do this, traveling across the continent multiple times a year, multiple years. It's a dumb way to operate. I mean, obviously people do it all the time, But you know, you could do an undergraduate project or a master's or a PhD thesis in California or nearby and really have a pretty meaty problem to chew on with some really nice tools to use with it. So finally, acknowledgments. My colleague Sasha Stefanovic basically bailed me out when I ran out of money for my graduate students and has been, sort of a molecular guru for the students, and the postdocs whom we co-supervised. We also got funding for what is this? Hawthorns as a new Canadian agroforestry crop for functional foods and nutraceuticals. Um, hey, we were in the money for a while and we got, that's where Jen Coughlin's work got paid for. That's where Nadia Talent and Mehdi Zarai's postdoc work got paid for. It was the gravy train for a short while, and we did a lot more than I've shown you. The Louise Hawley Stone Charitable Trust bankrolled getting Jim Phipps's collection, herbarium collection, to the ROM. The ROM Foundation provided peer review grants. I can't say enough about my colleagues at U of T. And here are some people who I really owe. Jim Phipps, Masha Kuzmina at the Barcode Institute in Guelph, Ontario, the late Rhoda Love, Peter Zika, and my former postdoc, Mehdi Zuray. And of course, my wife, who puts up with all this nonsense. Thank you.
That was great, Tim. We have time for questions. So feel free to unmute yourself or let's see, I'll do this so that I can see people if you've raised your hand. Better to start out by unmuting yourself.
Yes, Mark, unmute yourself.
Can you do it?
Actually, I don't even know what a thumbs up means.
Well, it might be good job. I should be able to unmute you.
Yeah, great job. Thank you for the talk. It was incredibly complex.
Sorry?
Great job. Very well presented, but of a very complex situation.
Thank you.
I want to remind people before anyone starts moving on that the next seminar is going to be on the 13th of February. It's Jesus Martinez Gomez at UC Berkeley, and he works on inflorescences. So Peter, do you want to go ahead?
Unmute yourself?
Yes. Thanks so much for the talk. Do you have a notion how unusual this degree of evolutionary complexity is? Is it a kind of a specific creature of the unusual requirement for fertilization of endosperm and for apomixis in this group?
Well, okay, first thing I should say that in parallel with the work that we've done, there's a group. It's now kind of they folded their tents, so to speak. Chris Campbell, who was at the University of Maine, led a group of students and others who worked on Amelanchier. And they a similar kind of situation as far as gametophytic self-incompatibility on the one hand, and the breakdown of self-incompatibility with polyploidization. The balance of hybridization and it's the same kind of complexity, but different. And then in Europe, there have been people working on what has been called sorbus, probably 5 different genera plus nothogenera. But there's been a great deal of work done on sorbus with the kind of tools that I've just described to you. There have also been people working on Crataegus in Europe, and with this again with the same kind of tools. Sorbus, Cotoneaster has been studied. All of these large quote unquote malloid genera share this kind of complexity. And those of you who know the Rhozoideae, things like Potentilla, Alchemilla, those genera, the Rosaceae are really chock full of this stuff. And I really don't know anything to speak of about those other genera in the Rhozoideae, but I don't think it's, well, pseudogamy, the pollination requirement, the requirement for endosperm fertilization, I think makes it a really powerful evolutionary motor for generating variation, because on the one hand, you can throw out all the stuff about species. Oh no, hybridization is bad. Well, no, hybridization is really fantastic. It's just a more extreme form of outcrossing than simply what you're used to with in diploid populations. And so what we see is that there are these genera. I'll throw out again Alchemilla and Potentilla, these malloid genera, Cotoneaster, Crataegus, Sorbus, Sensulato, and so on. All of these guys, if they have the capability for gametophytic apomixis, they can generate variation hand over fist and variance hand over fist, some of which will be cloning themselves up. Others are diploids like Crataegus monogyna. It's a diploid, but it hybridizes like crazy wherever it gets the opportunity. So yeah, there's a lot of potential for complexity.
So Andy, did you have a question?
Yes, I did. My question relates to Amelosorbus, which
Mhm.
is the hybrid between Sorbus and Amelanchier, which has a fossil record extending back 40 plus million years. And it made me think that, at least in that case, this hybridization can be persistent over geologic timescales.
Yeah, I mean,
So what I was wondering is, have you been able to, or do you know if it is possible to identify genetic domains or some other genetically or some other identifiable connection to the morphological differences that you are identifying between these different hawthorn groups? Can that be used to understand why it is that you are able to get these identifiable taxa, let's call them, persisting over evolutionary time despite rampant widespread hybridization?
okay. Amelosorbus is a name I have heard, but I didn't know that it had a fossil record.
Devoran Pig, Okanagan Highlands, Republic Flora.
I'll go back and look. I mean, I know that some of the Devoran Pig papers, and I've talked with them and enjoyed talking with them, but we haven't explored that, and so I don't know anything about that. I don't know. I mean, one of the arguments, I mean, that people have made is that it's the diploids that get through the really, really tough times. Like notionally, the survival of diploids south of the glacial margin suggested that, maybe the glacier, wiped out all kinds of diversity. And really, it was mainly the diploids that survived and were able to go and recreate that diversity. I don't know. What I did do was I worked with people in my museum and at the University of Toronto to go and we have a really nice Cretaceous collection at the Royal Ontario Museum now. And we also have a soft X-ray machine. And I was able to have money to pay somebody who knew how to run the X-ray machine and develop the negatives to go and get leaf X-rays showing the venation in really great detail. And I keep pushing that at Melanie DeVore and Kathleen Pig, to say, look, you find leaf fossils. Here is a database to use in trying to be sort of honest about identifying things as particular species, whether modern species present 20 to 40 million years ago or not. I don't know. But the point is that we're just not very far along with the kind of work that you suggest could and should be done.
In terms of what Kathleen and Melanie do, they're usually working at the genus level in terms of identifying what species they have. Is that they're able to say that there are 3 species in this flora, but in this fossil assemblage at this time period but they can't connect one flora that is, say, 40 million years ago to another flora 35 million years ago and connect the same species. They can connect genera.
Right. Well, one of the things that's pretty conspicuous, when I was doing all this stuff, I would periodically dip into the, sort of compilations of fossil taxa. And, the fossil hawthorns were always something that looked like a European hawthorn, like Cretaceous monogyna. It's kind of like, oh yeah, that must be a hawthorn. Everything else is, I don't know, malice or some damn thing. But hawthorns have have loby leaves like Cretaceous monogyna.
How old were the fossils you were looking at?
I think Eocene.
Okay, so it is possible that you've had continental rearrangements over that time period.
Oh yeah,
climate was
absolutely
really different in the Eocene.
No, for sure. I mean, the paper that was the publication for the X-ray database on Morphobank is a little paper I think it's in Phytologia that Roman Ufimov did with me. He's a Cretaceous taxonomist, capital C, capital T, all serious, not hand waving at all. And we collaborated on this paper in order to talk about the subgeneric classification. And one of the things that we wanted to do was to look at a polar view from 40 million years ago polar view of the continents 40 million years ago and superimposed not just the distribution of the subgenera, but also their relationships. And, there's every reason to think that there were groups across what is now the Atlantic Ocean, that some got left behind, some, died out. And similarly across Beringia with these black-fruited things, the closest relatives of the black-fruited things are in Eastern Asia.
So, there's one question in the chat that I thought we'd get to before we close out,
Okay.
which was just Marianne McCrary asking if you use the Cretaceous occurrences that are in the CalFlora database.
No, because I was young and stupid, and by the time I discovered CalFlora, I had also gone and borrowed a lot of specimens.
Okay.
And
You might have some more recent things.
absolutely. Oh, and in fact, I don't remember the name of the person with whom I was corresponding back in a couple of months ago. But one of the things about looking at CalFlora and looking for pictures, okay, so I volunteered. I'll go and work with Jim Phipps to update the Jepson treatment of Cretaceous. Fine.
Ooh.
So I go looking for pictures, and what do I find? But I don't remember which species it would have been. But the point was, nice black-fruited hawthorns from way over to the eastern side of California, northeastern side of California. So absolutely, I think, honestly, I think I'd rather do it visually with the CalPhoto database, or CalFlora. I mean, it's had one world enough in time.
Right.
But that's what I think that that students, looking for a project, just a bunch more tools ready and waiting for them to take advantage of.
That's great. So get the word out, everyone who's still on the Zoom. Tell your friends. Lots of work to be done, and lots of potential here. Thank you so much, Tim, for this really interesting talk and for encouraging people to get working on some of these questions. Everybody remember that we've got the next seminar on 13th February. Jesus Martinez Gomez from UC Berkeley, and tell everyone to get their abstracts in for the graduate student meetings. Thank you so much.
Well, and for my part, thank you guys very much, for offering me the opportunity and for taking part this evening. Thank you.
Pleasure. All
Okay.
right.
Bye now.
Encourage students
Bye.
I would also encourage students to participate.
Good.
I attended the first one.
Yeah, I organized
ago.
the second. I think the second one was at Berkeley, and I organized that.
Well, this first one was at Rancho Santa Ana.
Yep.
Yeah. Okay, guys. Good night.
Okay.
Bye now.
Bye.